Welcome everyone. This is our actual inaugural lecture series at the OSSA, the Ontario Science Students Association. And for our first lecture, we have Dr. Ray Truant, actually who had taught me a course in, was it already 2 years ago at this point? I'm pretty sure it was Biochemistry 3CB3, Emerging Discoveries in Biochemistry. It was a quite interesting course. I enjoyed it very much. And Dr. Truant is currently a professor in the Department of Biochemistry and Biomedical Sciences and was chair of the Scientific Advisory Board and board officer of Huntington Society of Canada from 2007 to 2021. His research group is multidisciplinary and strives to develop a therapy for Huntington's disease by any means necessary. The Truant Lab is in an academic setting, but highly collaborative with the pharmaceutical industry and biotech partners, as well as their clinical collaborators, Dr. Mark Gutman and the Center for Movement Disorders in Toronto and Toronto Western Hospital and the University of Toronto, as well as Dr. Mark Tarnopoyski at McMaster's Neuromuscular Disease Clinic. The Truant Lab has current projects in Huntington's disease, SCA1 and SCA7, and is moving towards elucidating universal mechanisms in age-onset neurodegeneration. Again, quick reminders, this is recorded. I will be putting links in chat for other weeks if people are interested, but without further ado, Dr. Truant.
Okay, so thanks, Izzy. Thanks to the OSSA for this invitation. And one of the reasons why I wanted to give this talk to this group of people is because during lockdowns 2020 onwards, we actually managed to get something really big done at McMaster that impacts the whole province and research throughout the entire province. So we formed this organization and facility called the CALM, the Center for Advanced Light Microscopy at McMaster. I'm just going to show you what that is. This is kind of a unique facility located in the Arthur Burns building on the campus at McMaster, and it's a truly centralized facility that involves 3 faculties. So health sciences director, which is me. We have an overall director, which is Jose Moran Mirabal, who's in science and who's a materials scientist, a chemist, and Chin Fang in engineering, who's an engineering expert. And throughout the process of setting up this facility, we not only managed to get it built during the lockdown, which has really been crazy, but it got done. It's several thousand square feet. We hired a real rock star as a director, João Pedro Bronze de Firmino, great name. He's trained all over Europe, and he's training people within the facility, and he's the facilities manager. And we immediately hired Mohamed Babi as a postdoctoral training person as well. So we have a lot of hands-on expertise in this facility and millions of dollars in brand new microscopes that we've just installed in support from the university, support from the province, and support from the federal government. We're also directly connected to the Canadian Centre for Electron Microscopy. The facilities are mixed with each other, so people can come in and get solutions and imaging from really advanced fluorescence imaging, mostly biological based, to atomic force microscopy, to electron microscopy and correlative light electron microscopy. One-stop shop. And it's primarily intended for the people at McMaster, but it's open to the entire province, and anyone who wants to come here can get trained and use the facilities. Okay, so my lab has been focused for over 20 years on the problem of neurodegeneration. It's a huge problem, right? 50% of the population over 65 is at risk. We've never had an older population in the history of humankind on this planet. So we have a record number of old people on the planet, and half of them over 65 are going to see some form of neurodegeneration. Massive costs. The only estimate I could get was back in 2017 of about $275 billion a year in the US only. And then there's all the burden of caregivers that we really don't count on this. And anyone who's had some form of neurodegeneration and a senior member of their family knows what I'm talking about, right? It's often put on other members of the family to take care of these people when they're at the stage where they need help. we need to figure out a solution on all of this and some therapies, because if we don't, by 2050, it's estimated that we'll basically bankrupt all the medical care systems in the world. There will be more people in 2050 in China with Alzheimer's disease than there will be people living in Canada. So the scale of this is quite huge. And I wish I could say governments are being proactive on this, but governments are never notoriously proactive on medicine. So hopefully we can get to some real solutions. The biggest problems that we have right now is we don't have any way of treating any form of dementia: Parkinson's, Alzheimer's, frontal temporal dementia, et cetera, et cetera, et cetera. There's nothing out there. A lot of this has been tied to some long-standing problems. One of which is there's often people, they say the word dementia, and there's an implied single mechanism for all of dementia. The reality is diseases like Alzheimer's and Parkinson's disease are not diseases with a singular cause. They are the result of probably hundreds of different causes. And in fact, they may be 40, 50 different diseases with a common output. So the analogy I make is very similar to the way we talked about cancer in the 1960s, where, cancer was one disease and let's cure cancer, right? War against cancer. But by 2022, we have had great successes in cancer. When we parsed it out to well over 200 different diseases, and some of which we've had great success with and others not so much. So the other big problem they've had in this research is that it's really, really been stalled intellectually and scientifically on one hypothesis, the amyloid hypothesis. And this was presented in a famous paper in 1992 in Science by Higgins and Hardy. That's John Hardy there in the picture, who's at UC London. And they're basically, the idea is that these neurodegenerative mechanisms of disease are somehow related to some protein misfolding hypothesis. In the case of Alzheimer's, it's things like tau, beta amyloid. In the case of Parkinson's disease, it's Lewy bodies that are full of misfolded proteins. However, that was 25 years ago. 25 years later, we don't have a single therapeutic based on this hypothesis, not for lack of trying. Industry has estimated to have spent well over $4 billion to date. All the big companies, Biogen, Janssen, Roche, Eli Lilly, have had $200, $300, $400 million programs that have all failed miserably. The most recent failure is adamanecab from Biogen, also known as Aduhelm. Hugely controversial drug. We actually, in the US, the FDA actually approved it, but no other country did. I'll talk about that in a minute. So there are some big conceptual problems that we need to overcome to understand all of this stuff. One of the problems is what's called the Lamppost Problem, right? And it's an allegory for, the story that someone's walking at night and they're walking along a path and they've dropped their keys and they have no idea where their keys are, but the only way they can find their keys is if their keys happen to fall underneath the lamppost and where the light is. If the keys fell outside of that light area, they're never going to find them, right? And the lamppost problem is something that's often created by the use of model systems that are not necessarily the same or encompass all the aspects of human disease. So model systems can be great and get us very far ahead if we understand the caveats of those systems. The second problem we have is what it's called the silo problem. And this is where everybody is in their own little silos. They go to their own little meetings, they publish in their own little journals, and they don't talk to each other. So very recently, there's been Alzheimer's and Parkinson's meetings in the same place, the AD/PD meeting. However, people in Parkinson's and Alzheimer's don't come to Huntington's disease meetings, and they don't go to other neurodegenerative disease meetings, and they tend to stay in their own little bubble of information without looking at broader mechanisms in other diseases, and don't really take a comparative approach. So if they think they're on a pathway in Alzheimer's disease, they don't ask the question, is the same thing going on in Parkinson's, the same thing going on in Huntington's or ALS or FTD? The last problem we have is the best idea I can come up with is what I call the black turtleneck problem. There has never been more con artists and grifters in science than there is right now. And these people are heading biotech companies, and they're raising huge money in the stock market on what are really bad and poorly developed ideas. And this has really been choking out a lot of potential resources and research. This is a quote from a friend of mine, Mark Smith. He was the editor of the Journal of Alzheimer's Disease for a lot of years. He died in a tragic accident in 2010. Just before he died, though, he published this review about the amyloid cascade hypothesis and the fact that it continued to dominate Alzheimer's disease literature and grant applications. And at some point he said, you know, this hypothesis would be the envy of any religion, because people have now taken it on faith, and we now have second and third generation labs that have grown up on this singular idea. And he asked the question of whether this hypothesis is too big to fail. This was 11 years ago. So these were people a long time ago, decade ago, that were raising red flags on this problem of a singular hypothesis in this disease. The main problem with the amyloid hypothesis is that you'll see all kinds of images like this on the internet. You'll see all kinds of medical experts talking to the media who basically get it completely wrong. They tell you that Alzheimer's disease is caused by amyloid. But if you go out and randomly scan the brains of a whole bunch of 80-year-olds, you're going to find brains full of amyloid, and they're not going to have any neurodegeneration. And you're going to find brains full of amyloid where they do have neurodegeneration. And you're going to find no amyloid and neurodegeneration. There's never been a direct correlation. It doesn't say anything. So if we can't scan someone's brain and say you're going to get or you do have Alzheimer's disease, you really can't tell if they have Alzheimer's disease until they clinically get it. And then you do a postmortem in their brain. So the biggest problem here is this Alzheimer's and amyloid and a lot of these other diseases where we see misfolded proteins, it's only correlation. Correlation is not causation. Correlation is not mechanism. What we really lack here is a mechanism of disease. The other problem that we have is Alzheimer's in particular is the golden ring of the stock market. If someone makes an Alzheimer's drug that works, like really works, it's estimated that drug over the 15-year period of the patent could be worth as much as $1 trillion. This says that these companies have to have programs on Alzheimer's disease, whether they're serious about it or not. Then what happens is you get people who play the stock markets, because these are all publicly traded companies. And so you get all kinds of things like these news stories that appear. Here's one from 2021. Lilly hails Alzheimer's win with donanemab, boasting significant slowing of decline. No, it's a year later and nothing has happened. But meanwhile, people have lost $24 billion in this stock. It's a classic pump and dump, right? So people are like, oh, look, we have something that could be a cure for Alzheimer's, and everyone goes in and invests, and then everyone loses all that money. So when you're looking at research budgets on neurodegeneration worldwide, where is your $24 billion better spent? On the stock market or in actual research? It's a tremendous amount of money, and it's going to all the wrong places on this problem. So Biogen is the latest huge failure on this drug called Aduhelm, which was FDA approved, but is essentially worthless because despite the fact that they got it approved by the FDA, there's no evidence this drug actually works, and there's a lot of severe adverse effects in patients. Enough that major healthcare chains in the United States are refusing to give it to patients. They said it's just not worth the risk. And other agencies across the world have looked at the data from Biogen and concluded this drug does not work. So the EMA withdrew it. And the Canada Health Agency, we did not approve this in Canada. There's zero evidence this drug works. Why the FDA approved it? Nobody knows. But it is currently a subject of an active investigation by the US Congress. which I think will be interesting. Then there's other problems that have appeared recently. If anyone keeps up with what's going on in science press lately, a lot of the major papers that were published in the early 2000s supporting the amyloid hypothesis have now been flagged as having fraudulent data, what I refer to as Photoshop science. This was mostly done by a few people who, do this on a volunteer basis and look at papers and identify what they consider to be strange figures, and then they work on it further and they figure out exactly how the figure has been manipulated. But there were several manuscripts in this time period that were obviously faked and in the process now of retraction. Well, that's a big problem because there's people who've built entire research programs on these papers. Now, if you guys want to keep hands on this kind of stuff as it's going down in real time, I encourage everybody to copy down this URL, pubpeer.com. This is where a bunch of nerds, such as myself, will go on there occasionally and discuss papers where people will be reading the papers and they'll say, this data looks a little fishy. But they're not experts. So they'll go out and they'll have a discussion forum where they'll say, look at the figure in this paper. Do you see anything strange about this? And you will get some real imaging experts. I mean, these guys are, pi mole experts. They come in with pretty sophisticated algorithms. And it is amazing what these guys can detect people have done to generate a fraudulent image. And these guys have triggered more retractions in science than anybody else. And it's free to look at, anyone can join. And anyone can be involved in the discussion. But you also have to remember that the discussions around these papers are extremely respectful. There are mistakes in science. That's not the same as fraud. And what they're concerned about is actual people trying to sell misinformation in the press. So when I look at the problem of neurodegeneration, it seems almost insurmountable. And back when I was a postdoc and starting my lab, I was like, people were telling me, oh, you should get into Alzheimer's research. The problem with Alzheimer's research is that I saw it like this. It was a disease of many, 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 many causes at the top of this tree and many, many effects at the bottom of the tree. The effects are the basis of a lot of neuroscience research, right? Where people are understanding all the various ways that neurons die during this neurodegeneration. The causes are likely very many. And we don't even know what all the causes are. We have the first clue what causes most of Alzheimer's disease. We have barely 0.1% of the population has a genetic component to it, right? So the way I looked at this was, if I'm going to understand this problem, I need to eliminate half the problem. Get away from all these unknown causes and go to a set of neurological disorders that are caused by a mutation in one gene. Monogenic disorders, right? That gives us our cause. Now I can work in a population of people that have one gene, one mutation, one cause of disease. We can predict if they're going to get sick decades ahead of time. And when they do get sick, we know exactly why they are sick. And you cannot do that for Alzheimer and Parkinson yet, right? So we're getting rid of all of these causes problem, and now we're focusing on the effects. And when we focus on the effects, I don't really care about the branches in the tree. Those are things that, from a therapeutic perspective, are going to make people's lives a little better, maybe, but I really care what's going on up here in the tree trunk and what are the root causes of these diseases. So I focused on Huntington's disease for a few reasons. The main reason is it's a monogenic disorder, and the second reason is it's a very well-developed research community. It was originally described by George S. Huntington, who's an MD, and someone who, like every good MD in his era, a lot of these people inherited their practice from their parents. So he was like third generation of his family, with the same families that he's been treated, that have been treated for generations. And there was a genetic founder effect in this part of New York where his father was treating a lot of these neurological cases. And then his son was treating the sons and daughters of the people that were involved in these neurological diseases. And he gave this fantastic presentation, 1872, at the age of 22, which is incredible. where he understood the hereditary nature of Huntington's disease, and that, you know, the thread, if it's broken in the families, the great children and the grandchildren are the original shakers, as they called them back then, may be rest assured that they are free from disease. He understood the genetics of disease almost 100 years before Watson and Crick and before modern genetics, which is incredible. So he deserves a name on the disease after him. One of the areas where there's been a lot of focus on this due to an isolated genetic founder effect was Lake Maracaibo in Venezuela. 1 in 120 people have Huntington's disease within this community. And this led to the cloning of the gene, coordinated by Nancy Wexler at Columbia, who's very famous in this field and in fact written many books, and a couple of Canadians working down in the States, Marcy McDonald and Jim Gazella, who are now at MGH Harvard, but actually originally started these projects at the University of Toronto when University of Toronto had a huge genetics department that was fantastically successful in that time. That's the era that I was at U of T. It was exciting. Like every month there was like a new gene cloned, and Nature would come in, and the press would be all over the place. And these were guys who were profs in my department. It's pretty exciting. You know, of course, they all got recruited away to the United States one after another, but at the time, Toronto was a world powerhouse in this field. So it's an age onset neurodegenerative disease, right? I've already explained it's one gene, one cause. It's a huge protein. This is the big problem. 3,144 amino acids. It's one of the largest proteins in the human proteome. There's a magic number. There's CAG repeats that encode glutamine in the protein. If you have less than 37, you're never going to get Huntington's disease. If you have over 37, you will eventually get Huntington's disease. But this only actually predicts 50% of the age of onset, which I'll talk about. So the pathology is restricted mainly to the striatum, so the base of the brain, the part where the upper brain communicates to the spinal cord. That part degenerates, which is one of the reasons why you get this choreic or movement disorder in these patients. And you get a lot of dysfunction before you actually get death. In fact, the first symptoms that hit these patients are typically, severe depression, mood disorders, way before they have any physical movement symptoms. And it's become very popularized by the people who have had it in the past. So for those of you who know, the basic folk music in the 1950s, 40s, in the United States, the Guthrie family was famously affected by Huntington's disease. It killed, Arlo Guthrie, and it's killed many of the Guthries in the family. It was made famous in the TV series House, with one of the characters on the show who was playing the part of a doctor who was a Huntington's disease carrier. So the approaches that people have taken over the years have been really widespread. My lab's approach was one of the first labs to do live cell imaging of cells and neurons while they were undergoing death due to Huntington's disease, using various methods of fluorescence microscopy. But we have over 12 model systems in rodents of various types. All the proteomics has been done. We have all kinds of cell models, including a recent cell model developed by my lab directly out of HD patients, human cells that are not transformed. We've done systems biology, all the omics you can think of, and a very important project that initiated actually, but was published first in 2015 with clinical studies with 16,000 people enrolled, which is incredible for a disease of this incidence. And we've done genome-wide association studies of over 9,000 patients. And now, unfortunately, because of an amyloid-like hypothesis where people saw protein misfolding in the disease, most of the models of this disease, in fact, remove 33 out of the 34 exons and only use one exon of the protein. So for a 3,000 amino acid protein, they use 81 amino acids. It's really gone nowhere. But we're not doing that. Right? So GWAS was very interesting. We asked the question, using the power of genetics in 9,000 people, so tremendous statistical power, what are the other genes that define the differences between people? Because if two people have 43 repeats in their genes, in their Huntington gene, they can have an age onset as early as 23 or as late as 65. That's a huge difference. Why is that? So they went through and they did genome-wide association studies. Before this happened, the amyloid people were saying, oh, you're just going to find a bunch of proteins and enzymes involved in the proteostatic mechanisms, chaperones, things like that. Why even bother doing it? Regardless, they went ahead and did it. And it's very nice to see data this way because there's no bias in this. It's statistics. They just ask the question, what pops up? And to everyone's, well, not everyone's surprise, but a lot of people's surprise, not a single gene involved in protein homeostasis has any relevance to the age of onset or severity of Huntington's disease. It's all mitochondrial functions, mostly DNA repair and redox control. So something to do with oxidation and mitochondrial function, but primarily on the repair of damaged DNA due to oxidative stress. And this was absolutely not seen in any of these rodent models, but it's seen in humans where it matters. Okay, so this is what I tell people when they want to come work for me, right? I said you have a choice here, right? From the Matrix, blue pill, red pill. I'm not even sure I got the colors right on this, but anyway. So you take the blue pill, amyloid causes on neurodegeneration. You go off and you publish papers in, various specialty journals. You'll get a PhD. You might even get a grant. But you take the red pill and you look at alternative approaches to come work for me, because I have no interest in amyloid. So in 2022, all right, so that we're now, almost 30 years after Higgins and Hardy, the amyloid precursor protein, APP, is called the amyloid precursor protein. We don't even know what this protein does. Nobody really cared for about 25 years. There's some interest now in figuring out exactly what it does. But one of the main questions that I was asking from the beginning was, what does this Huntington protein actually do? Why is it important? It's essential in vertebrates. It's essential in all cells. Every single cell in the human body expresses this. It's expressed in all cells. There are no HTT, there are no Huntington null humans, right? You cannot make a human being without Huntington. Haploid insufficiency, so the loss of one allele, is an extremely rare situation. If there are any hypomorphs, another extremely rare situation, because I don't think most of these people would ever even develop, they get an onset neurological disorder that's pretty severe called LOMARS. So it's an extremely important protein in terms of general cell biology, which is why I find it interesting. And one of the first things that we characterized was that in the very first 17 amino acids of this protein, there's two important serines that are modified. And they're not being properly modified if there's polyglutamine expansion in the protein. And it's a classic biochemical problem. It's an amphipathic alpha helix. And if you put in a phosphate on there, it changes the shape of the helix, and it affects the ability of that helix to interact with lipids. And the lipids are in the endoplasmic reticulum. So this is what an antibody to total Huntington looks like. This is what an antibody to phospho-Huntington looks like. It's a binary switch. You get a phosphate on the protein, now all of a sudden your protein is in the nucleus and hovering around chromatin. When it's not phosphorylated, it's sitting down in the ER. And what it's doing out there is it's sensing, it's looking for trouble. It's looking for elevated levels of reactive oxygen species. And if that happens, it gets off of the ER and books it into the nucleus to address the problems. And we've looked at this through a variety of methods and a variety of technologies. So this is something called a camelid chromobody. So this is a piece of an antibody from an alpaca. We use these guys because they have an unusual immune system. They make these tiny little antibodies. They don't have heavy and light chains like other mammalian species do. We fuse that to a fluorescent protein, and we now have an antibody that can highlight endogenous protein inside of a living cell. We can then stress the cells with an oxidizer like potassium bromate, which affects nucleic acids, and we can see that the protein moves from the cytoplasm and starts accumulating within the nucleus. And we've done a lot of biochemistry to explain that when this signal, this again, 17 amino acids of a 3,000 amino acid protein, so this is really the tail that wags the dog, will change how this protein can localize, if it's just oxidized or if it's phosphorylated. The single amino acid is really critically important. So we wanted to carry this further and see, can we find small molecules that fix the problem? The problem is the mutant protein is not getting phosphorylated the way it should be. So the question is, can we find a small molecule that restores this problem to normal, right? And to do this, we hit it with a lot of technology. We did a lot of microscopy and development in my lab, and the use of what's called machine-based sorting. And this is why we're doing machine-based sorting. There's a really brilliant guy named Robert Murphy at Carnegie Mellon, who proved that if you get 3 experts to look at an image and score it based on what they think they see, they're only at best 70% accurate. But if you train computer algorithms to look at many aspects of an image to get what you want it to see, you can get almost 100% accuracy in those images. And I'll give you an example of this, that humans are not the best people to be looking at microscopy images. So I'm going to flash up an image in a second, and I want you guys to score in your mind what you think you see in this image, right? Here it comes. Boom. All right, you're looking at this. Now, I know I could poll you guys, but I know half of you are going to say that you've got this is an image of someone in a parka facing away from us. And about half of you are going to say, no, this is the image of a woman's face looking at us. It's the same image, right? It's the same pixel set with 2 completely different interpretations. And this has to do with the way that humans process bias. We're really bad at it. Turns out pigeons are better than humans at this. All right, we didn't use pigeons. We used computer algorithms instead. So we had a random library of compounds that are all known bioactive so that we know they do something, hundreds of them. We randomly captured images on a robotic microscope, and then this gets pushed into something called unsupervised machine sorting. So essentially, we use principal component analysis to just answer the question, which compounds make a difference to the image set that we have here? So front to back, no humans are involved in scoring any of this. It's all done automatically, right? And what it gave us was this. Well, most of the compounds don't do anything. They cluster here in the middle, and they're really not doing anything to our data set. We're looking at our signal here is how much phosphohuntington we have. Other compounds are doing something, but they're doing something different here than they are here, and very different than this guy number 50 here. So turns out these guys all ended up being antioxidants. So they would reduce the phosphorylation of Huntington. The other ones turned out to be inhibitors of a known pathway involved in NF-kappa B signaling, but we had already published on this previously. But this guy was very different. It was not here or there or there. It's just different. And it turns out it's an interesting little molecule called N6-furfuryladenine, also known as kinetin. And we showed now that if we take this compound and put it back, we can restore the phosphorylation on the mutant protein. We can fix the phosphorylation problem. All right, get it in the mouse. So we used a mouse model with Simonetta Scipione at the University of Alberta. And we used multiple doses. The mouse model was actually developed by John Roeder at University of Toronto. In a rather interesting but tragic story, Roeder developed this mouse model. Roeder was on my PhD defense committee, and he was at the Lunenfeld, worked there for decades, never told anybody that he had Huntington's disease. So he just, he made this mouse. He's like, I got a problem, I'm going to try and fix it myself. So he made a really good mouse model of this disease. So we treated that mouse model of disease, and we got the body mass was fixed, better rotor rod activity. All the movement disorder parts of the animal were fixed with the treatment of the animal, and lower anxiety and low dose oral. Interestingly, when we restore the phosphorylation of Huntington, we get rid of these protein inclusions everyone's on about, even though we don't have something that affects proteostatic pathway whatsoever. So we published that in PNAS, and that's now being developed by a biotechnology company in the United States, in San Francisco. We've also developed these new cell lines. These are coming directly, fibroblasts directly from patients. But instead of typical cell lines that transform and immortalize cells, we use HTERT, human telomerase enzyme, to immortalize the cells, but not transform them. So these are not cancerous cells. These are cells that simply will live on forever. And so why do this, right? Why not just use, like, HEX or HeLa cells? And the reason is they have incredibly stable genomes, unlike HEX and HeLa cells, which are never the same cells twice. If you take a plate of HEX cells and split them, it's a different group of cells the next day. There's extremely unstable genomes. TERT immortalization gives us extremely stable genomes, but they give us cells that don't die. And as a side note to all of this, TERT enzyme was discovered and developed originally at McMaster by Cal Harley in this department in the 1980s. But he went on to the Geron Corporation. So with one of my now PhD graduates, Claudia Hung, she developed this cell line to show that there's phenotypes that we can see in the skin cells of HD patients without the need of any model system or fragments of the protein or any synthetic alleles or overexpression or any of that. We can see it. So if we can see a problem, we can try and fix the problem. And we can see what happens in DNA damage. So if we burn a stripe of damage onto the nucleic acid using a 405 nanometer laser, we can see endogenous Huntington piling onto that damaged DNA. It's responding to the damage. This is again using the chromobody in a laser stripe assay. And my research associate, Tamara Mari, went on, who's another U of T graduate actually. So she went on to show that all of the enzymes and players that are involved in the processing of this N6-FFA small molecule to give the phosphorylation of Huntington all physically localize to DNA damage that we induce by burning a stripe of damage using an ultraviolet laser onto DNA. So our questions were now, well, how is this Huntington protein seeing DNA damage? A few interesting things have come up recently. We recently solved the structure for the majority of the Huntington protein. And I recently bought a 3D printer. So I can print this guy out and hold it in my hand now, this complex of Huntington bound to another protein with 2 angstrom resolution. It gives us about 60% of the structure of the protein. The other 30% is actually unstructured protein. It flops around, and that's important. So we wanted to know how Huntington was seeing damage. Well, when we think about DNA damage proteins, one of the first steps that occurs in the response to DNA damage is an important enzyme called PARP1, poly-ADP ribose polymerase. Basically, it's an enzyme that sits down, recognizes the DNA damage, and then generates these long chains of ADP ribose. The analogy is here. Okay, so we have this boat, which is damaged DNA. PARP1 is sitting down on the damaged DNA, and there's DNA repair proteins floating around the nucleus, but they're not where they need to be. They need to be at the DNA damage. And so the way that they're recruited is PARP generates PAR. It generates these chains that fish in the DNA repair proteins and bring them to the damage, get the job done, right? So we followed along this pathway, and what Tamara discovered is that Huntington contains a PAR binding motif. It contains a protein motif that can recognize poly-ADP ribose. And it looks like one seen in other proteins, and it's absolutely conserved 100% in all Huntington species from mammals and vertebrates. Together with Rachel Harding at the SGC Toronto, by looking at the structure of Huntington, we can actually see where this PAR binding motif is, and it's sitting right out there on the surface, right where we hope it would be, right? We did not want to see it buried in the middle of the protein. And if we now treat cells with an enzymatic inhibitor of the glycosylase that chops up the PAR chains. So in other words, the PAR chains form, they're broken down. This happens on the order of seconds. It's an extremely fast response. If we stop it though, and leave the chains long by inhibiting the glycosylase, and then we image Huntington, we get these stunning images where you can see in a mitotic cell, right? So there's two spindles here. That's what those bright spots are. We can see our phosphohuntingtin just painting the chromatin, showing this association with chromatin. And then we wanted to ask the question, well, what's going on in the disease? Is this real? Like, who cares? So the protein binds poly-ADP ribose DNA repair. What is this relevant to disease? So we got cerebral spinal fluid samples from quite a few HD patients. These were harvested at UC College London by Dr. Ed Wild and analyzed by Lauren Byrne, a PhD student in his labs. Now she's a postdoc, she graduated. And we used an assay developed by Ted and Valina Dawson at Johns Hopkins, who are very famous in the Parkinson field for demonstrating that there's a problem with PAR levels in Parkinson disease. So we used the assay developed by Tay and Cam. And what we could see is, what we expected to see was that there would be hyperparylation in HD, the way there is in Parkinson disease. What we got was the exact opposite answer, which really confused us. So not only is there less PAR response in HD, but it happens worse in pre-manifest HD. So this is decades before these people are anticipated to get sick. Something is going on very wrong and very early in the disease. So we've now branched out worldwide with a lot of collaborations. One of the most interesting ones lately is with a company that's formed in Krakow, Poland, with Magda Corden. That's Magda there in the picture. So they've used a technology they refer to as stride labeling of DNA damage. So this is a mechanism where they directly use a chemical reaction to put a fluorophore, and they can specifically see single-stranded breaks of DNA or double-stranded breaks of DNA. And so we had them, try this assay out in our HD cells out of the patients, and we got two really cool answers. One, the problem is more double-strand breaks in Huntington's disease. And the other interesting thing is, it really only happens in cells that are not dividing. If cells divide, they can repair the problem, which now we take a step back into patients. If someone is born with 43 repeats and you look in their blood cells, they're going to have 43 repeats in their blood cells their entire life. But if you look in their brain, you're going to see those repeats expand out beyond hundreds. There's a somatic expansion event that doesn't occur in replicating cells, but does occur in non-replicating neurons. And we can see this with their technology with this group in Poland. And we're continuing on to work with them. Then, of course, we had a surprise. Now, you guys, one of your speakers in the next coming weeks is Sarah Andres, who's a DNA repair specialist whose lab is literally down the hall from mine. And we asked her, it's possible that we might be able to see Huntington, pure recombinant protein, actually binding to DNA. Can you take a look for us using atomic force microscopy, right? So it's kind of a misnomer. It's not really microscopy because there's no optics involved. Atomic force microscopy is where you drag a probe over the surface of mica. Mica is almost perfectly atomically flat. And you end up with a bump map of single molecules that you can see in this kind of space. Tremendous resolution, nanometer resolution, and you're looking at a chip size here of 500 by 500 nanometers. So we can see one molecule of DNA, and we can see multiple molecules of Huntington. So every one of the bumps in this image is a molecule of the Huntington complex with a protein called Hap40, and we can see it binds to those double-strand break ends, and it binds within the DNA. And of course, because I can't leave things well enough alone, I figured out a way to get this type of data and actually 3D print it. So this is the kind of thing that I now bring with me when I go to meetings. I can literally take the data and show people and say, here, look, hold it in your hand, touch it. And it's been an extremely cool technology. And the only reason why we would have done this is because we happen to have two devices sitting within the COM facility, And I happen to have some really good experts and know how to use this device. And I got the input from Sarah's lab. And she's probably going to talk about stuff like this during her presentation to you guys. Then we went on to look at PAR chains by the same method. Can we see poly-ADP ribose by AFM? And short answer, yes. You can see in the middle here is PARP, and here's all the chains of the poly-ADP ribose. Now we mix in Huntington, and we got a very surprising result. The entire morphology of the chains have changed. So when I showed you that video of that net being cast, it turns out the different types of PAR chains will attract different types of proteins. And Huntington is directly binding PAR to somehow modulate this. And this is where we have a lot of current interest. So we're now getting an idea at the trunk of this tree that our very first problem is aging. If we can, okay, so if we can stop people from getting old, fix the problem, but we can fix a lot of problems that way. That, of course, biochemically is reactive oxygen stress. We know that reactive oxygen is a component of all neurodegenerative diseases. It's elevated, mitochondria don't work as well, older people have higher raw stress. Why is it that some people can handle it and others cannot, right? But clearly that raw stress, what it's doing is DNA damage. And if that DNA damage is not properly fixed, depending on the subtleties of what that damage is and how it's not fixed, it can generate Alzheimer's, Parkinson's, FTLD, ALS. ALS is a very well known to be a ROS-dependent disease, or Huntington's disease if you happen to have mutant Huntington. And this is coming now out of the press from clinical studies. So in Norway, this group sees that they see DNA damage before they see the mitochondrial problem. So the mitochondrial damage precedes the mitochondrial problem. And other groups in Italy in clinical studies looking in peripheral blood cells out of patients are seeing that the DNA damage is the very first thing that they can see in Huntington's disease. And importantly, it occurs in prodromal HD. So back 20 years before we expect these people to get sick, when we go back now to our CSF data, we're seeing there's something wrong with the PAR response there. So this says this disease is starting in persons' 20s, not in their 60s when they go to clinic. And there's a lot of interesting biochemistry of what happens in the aging human brain. Turns out for those of you, I'm going to bring you back now to second year biochemistry. Don't freak out, but you got to remember glycolysis, oxidative phosphorylation, Krebs cycle, all that stuff that you forgot the minute after you wrote the exam. It's really important to all of this, because what happens is the human brain is not always good at producing the energy it needs to produce in an older person. So the brain will flip between oxidative phosphorylation to glycolysis, right? The burning of sugar to generate ATP. So in doing that, it'll quickly get that ATP that it needs. But the problem with glycolysis is it generates a lot of reactive oxygen stress and it's not very efficient. So you don't get a lot of ATP. So the brain flips back and forth until age 60 in normal people. At age 60, the brain knows don't go to glycolysis because glycolysis is going to cause more problems than you try and fix. But in AD patients, they keep flipping and they keep flipping. And if you look at the regions of where they're still flipping, these map perfectly to the regions affected in Alzheimer's disease, right? So there's this concept now that we have a stuck chronic stress response that we may be able to unstick with therapeutics. And there's a variety of clinical trials going on in this space for Huntington's disease. But 2 weeks from now, I'm down in Boston, chairing a session. And one of the things that I've been talking to with companies like Novartis is that we need to rethink how we're dosing these diseases. Right now, what we do, this is typical of any disease, right? Of any acute disease where we do nothing when the person's not sick. When they show up to the doctor and they have the disease, we then think about giving them a therapeutic. And this is how we trial diseases. We wait for someone to get Huntington's disease, we score them clinically, we give them a drug, and then we hope that it fixes the disease. Well, the problem is the reason why they have the Huntington's disease is those neurons are gone. So those neurons are not coming back. There is some neogenesis of neurons, but nowhere near enough to fix this problem. So the disease is not in these set time periods. It is progressive. It starts at a young age, back in teens, early 20s, and just gets worse until they're older, and then they have full-on Huntington's disease. So we need to stop thinking about putting the therapeutic here and start thinking about putting the therapeutic here. And the therapeutic is to reduce levels of the mutant Huntington protein. We don't want to do it too early because it's important for development. So if we give babies these drugs, we're going to give them low MARS. So we don't want to do that, right? We want them to get to development. It'll probably be somewhere around the time that guys start wearing these shorts. But then we'll carry that forward for the rest of their lives and give them low levels of drug, but for the rest of their lives. And presumably, we can then chemically mimic what we see in GWAS. We can affect the situation very slightly so that the real readout in clinic is decades. Because in genome-wide association studies, if we know what these gene modifiers of disease are and we look in the patients, there's very subtle differences between these patients, maybe 10 or 15% expression levels of the proteins or activity that will read out to a real 30 years in the clinic, which is pretty much a home run, right? If you can just push off the disease by 30 years, by the time these people are in their 80s, they're going to, die of whatever else the rest of us die of, which they're totally fine with that. And, so this is suggesting that we really need to think of Huntington's disease more along the lines, and I think a lot of these neurodegenerative disorders, along the lines of phenylketonuria. So whether you guys realize it or not, every single one of you has been tested for phenylketonuria. You get it as a heel prick when you're a baby. The minute you're born, they prick your heel, they take a little blood sample, they check for a few different things. One of them is phenylketonuria. Because they know that if you have PKU and you get phenylalanine in your diet, you're going to be in a huge developmental problem. But if you can avoid phenylalanine in your diet, you are not going to get PKU disease ever. So it's one of these things where we need to be on a preventative scale. Okay, so my lab is supported by a lot of grants, and I've got a list of the people here, and you can see we're very collaborative. We work with a lot of labs worldwide, a lot of agencies worldwide in Europe and the United States. My main funders are the Krembil Foundation in Toronto and the Canadian Institutes for Health Research, which have been supporting me forever. And now we have this awesome tool of the CALM, which was OIT and the CFI supported. I've got grants in the New Frontiers in Research Fund and the HDF, HDSA, also NSERC. My lab is an open science lab. So we actually, publish our data in real time in some cases. And we even have a knowledge translation site that one of my students started, Celeste Seward, called SCASource, which has become very popular and now ended up self-propagating into quite a successful knowledge translation resource for patients who have spinocerebellar ataxia, which is another monogenic disorder. So I managed to get this done with 4 minutes to spare. So if there's any questions, I can answer them, hopefully.
Yeah, thank you, Dr. Truant. I will probably read questions if people want to put them into chat if they don't feel comfortable raising their hands. So I guess we'll get started. So from David, knowing the hospital systems may not allow easy access to clinical testing, how should we go about to give younger generations to get HDD tested in their prodromal states?
Okay, so this is a different problem in Canada versus the United States. In the United States, genetic testing, you have to get it done on the sly. You have to get it done privately and anonymously, because you cannot let your insurance provider know that you have a genetic disease, or you're never going to get any kind of insurance in the US. It's a great system. In Canada, it's not a problem. So if there is a family history of Huntington's disease and your doctor is aware of it, we do a genetic test on these patients. And it's not one of these things where you give a blood sample and then someone calls you up. It's very carefully done. So you need to, if there's a risk of the disease in the family, You need to arrange for the testing, but these people also have to meet with genetic counselors, and in some cases have psychologists help them out as well. This is some people don't understand this unless they're in the situation, but being told that they are the person in their family that has Huntington's disease is just as traumatic as being told by some of these people that they don't have Huntington's disease. They get a lot of what are referred to as survivor guilt, right? They're like, why didn't I get this disease, but my brother or sister did get the disease? And a lot of them don't believe the negative result. That's extremely common. We have a genetic test. We can give it to someone with their own authorization beyond age 18. But it may surprise people that only about 20% of people at risk do get tested. And the reason for that is they would rather live with the uncertainty than live with a certainty. And I mean, you guys can think about this yourself, right? If someone were to tell you at 18, I can give you a test that says how you're going to die and approximately when you're going to die. Would you take that test, knowing that there's not much you can do about it? A lot of people are saying, no, I want to live my life with the same uncertainty as everyone else, where I can go off and get hit by a bus tomorrow, right? And they don't take the test. When these drugs do come on board, we've got a lot of clinical trials going on right now. I think that's going to change. If there's a situation where you can do something about it and get onto a drug, then I'm sure we're going to see a lot more testing. And we have various levels of help from charities, including the HSC, to get everybody tested who wants to get tested.
Yep, we got another question from Chris. A friend of mine and his sibling have Huntington's family history and have chosen not to get tested. If this were tested at birth like PKU, do you believe it would be something that people would still want to opt their children out of?
Well, so the situation that's different with PKU is, with PKU, I think any parent wants their kid tested because they know that if it does come back positive, there's immediately things you can do to prevent PKU from happening. But that's not the case in Huntington's disease right now. We don't have any way to treat these patients. So it's really up to the parents are going to decide, you know, if we do decide it's a good idea to test early, it's still going to come down to consent. Parents are still going to have to be able to make the decision to allow the test or not. This is not something that you want to, you know, push on to people without fully informed consent.
Yeah, if anyone has any other questions, I do have my own, but Dr. Truant, if we could drop your email, let me pull it up into the chat for people to have. But my question is, oh, we have another question from David. What are the remedies to reduce the oxidative stresses?
Short answer, there are none. I mean, we've been looking at this for 20 years, right? We know, you know, there's oxidative stress is a problem. Dysfunctional mitochondria with aging is a problem. People thought that the idea was that we take antioxidants, and the nutraceuticals industry and GNC and all these other various scam artists are out there selling you all kinds of berries and teas and juices and waters that are supposed to reduce oxidative stress. None of them actually do that. And it's very difficult to even know if something like that works. The other problem with attacking oxidative stress is there's a lot of critical signaling that depends on oxidative stress. So there's been a lot of examples of people who followed, you know, Linus Pauling in his semi-insane rants about vitamin C and hyperdosed on vitamin C, who ended up triggering their own death through gastrointestinal cancer, because they've completely messed up some signaling pathways that were critical anti-tumor pathways that were relying on ROS. ROS is not always a bad thing. There's critical signaling pathways that need ROS, right? So if we just, you know, sort of bathe somebody in an antioxidant, we could be causing more problems than we think. what it universally turns out to be true is athletically active people will during their athletic activity, due to their muscle burning of energy, they generate tremendous ROS levels. And that ROS level gets sopped up very quickly by a series of transient responses that take place as a result of exercise. Those responses don't take place in sedentary people. So this is where we see that, if you look at active people, they have much lower incidence of all these neurodegenerative diseases because they have an inherent ability to deal with high levels of reactive oxygen because of the pathways that are activated during exercise and that type of stress response. So, it's a universal message. Healthy people, active people are generally healthier for a lot of different diseases, not just neurodegeneration. They have less diabetes, they have a lot of problems less.
Yep. And I guess to end it off, unless anyone has any other questions, I'll ask my own. It seems like there's a twofold problem kind of with the localization problem because of it lacks being phosphorylated, and then also the PAR dysfunction and dysregulation. So then would it be better then to approach a method like CRISPR in terms of cutting out the mutant allele for, I guess, young adults once they've completed development and just having the regular Huntington allele left?
Yeah, so the drugs that are in trial right now fall into two categories: antisense oligonucleotides or siRNAs that are going to shut down the production of Huntington messenger RNA, and we're going to turn off Huntington. Or we have these small molecule compounds that I've been working with Novartis on that cause a missplicing problem, and they cause the RNA to get degraded along the nonsense pathway. The net result is the therapeutics that are in clinical trial right now are to reduce Huntington levels, total Huntington. Now, I told you it's in every single cell, right? And it looks like it's an essential protein, but I was completely knocked off my chair when I saw the results of the phase 1 trial data. We could reduce Huntington by quite a bit in these patients, and we saw no effects. And we see no effects in our tissue culture cells as well, no negative effects, which says that there is something redundant with Huntington, which is good news, which means that we have molecules that can reduce Huntington levels, and we need to figure out how to get them into the brain better. But even if we don't get them entirely localized within the brain, but to the rest of the body, it's not that bad. So we can think about now, given the complete lack of toxicity, I'd say this gives us rationale to go into people much earlier, and then look at the. These are kind of concepts that could take us a decade to figure out, right? We're going to have to dose people for a decade or more. We're going to find out if this works or not long after I'm retired, not during my career. It's kind of crazy. This is the nature of these long, drawn-out diseases where you pretty much have to pass the torch on to the next guy because there's not going to be a simple answer in a matter of 5-year clinical trial. It's going to take us decades to figure out if it works or not.
All right, we have one last question. I guess we'll get to shove through and then we'll end it off. Has there been other relationships developed between your research findings and other neurodegenerative diseases?
Yeah, so I mentioned in there that we're working with a group at Johns Hopkins. They have seen that PARylation is off in Parkinson disease, and they're working on disease therapeutics in that way. It's very interesting. The PARylation has been seen as incredibly disrupted in Alzheimer's disease 1993. But the problem is that back then, everyone jumped on the amyloid hypothesis and they left the DNA damage hypothesis to the wayside. Nobody worked on it. Now there's a tremendous amount of interest in going back. But it's really the danger of following one hypothesis blindly, right? They ended up, they had a fork in the road. Do we work on DNA damage? Do we work on amyloid? Everyone went the wrong way. They went to amyloid. Now we got to go back to the fork in the road and go down the DNA damage pathway and see how that works out. So yeah, there is a lot of these pathways that are affected in all the monogenic disorders. So all of the CAG expansion diseases show some defect in DNA damage repair, and it's definitely a problem in ALS.
All right. I'd like to thank everyone once again for attending, and I'd like to give a big thanks to Dr. Truant for actually giving us this wonderful lecture.
Thanks.
Hopefully, if you guys are in second year or third year, you would be able to take the course that Dr. Truant teaches. It's very fun, very interesting. And the last thing I'll be sending in chat is a quick feedback form for us to know what you guys enjoyed, what you guys want in the future, so that we can keep improving this lecture series as we go into in our next weeks. Thank you, everyone.
And some of you are going to be going off into grad school, and just remember that CALM is a universal facility for anybody at any university. You don't have to be at Mac. We're here if you need us.
Thank you, everyone. Cheers.